Bon, ça me fait plaisir de vous accueillir ici au séminaire en informatique cognitive et surtout d'accueillir Olaf SPORNS. On parle du parrain de l'IA, des parrains de l'IA. Alors, un des trois parrains habite ici à Montréal, mais il y a aussi les parrains du Connectome et c'est Olaf SPORNS. Donc, je suis vraiment enchanté qu'il est là. Je vais vous lire un petit peu de son résumé. Non, pas de son résumé, de sa bio. Il est professeur distingué à l'Université de Indiana, connu par ses travaux en neurosciences, en particulier dans le domaine de la connectomie, l'étude de la connectivité cérébrale. Il a contribué de manière significative à la cartographie du connectome cérébral en soulignant son rôle dans la cognition et le comportement. Il est auteur de Networks of the Brain, et cofondateur du projet Connectome, the Connectome Project. I'll quickly say some of this in English, because those who are the Anglophones have probably seen the English app. I'm delighted to welcome Olaf SPORNS to Montreal to speak about AI and the connectomy. Olaf, do you pronounce it connectome or connectomy?
Connectome, just like genome.
Okay, cool. AI and the connectome. We have here in Montreal the godfathers of, one of the three godfathers of AI, but Olaf is surely the godfather of the connectome. And I don't want to take up his time. He's asked that you let him give his talk without questions, and then he'll devote himself to the questions. So with that, I pass it to Olaf. Welcome to Montreal.
Thank you so much, Stephen, for the kind invitation to speak. And the topic of today's talk really was suggested by you, Stephen, and I eagerly accepted the challenge of talking about the connectome and AI because I had been thinking about this for a while, and there are some interesting links and distinctions as well. But a disclaimer first, while I know, I think, a fair amount about the connectome, I am much less of an expert on AI. So my thoughts on AI, mostly at the end of my talk, are meant as discussion starters. So we'll see where we end up. In neuroscience, networks and network science approaches have really made enormous progress in the last decade or two. And I use these iconic images on this slide just merely to make the point that across a wide range of systems, from invertebrates to mammalian brains to non-human primates to the human brain, and across different measurement techniques, connectome networks have now been mapped very extensively, and the research project of mapping brain networks has become front and center in neuroscience. And I want to give you a brief overview in this talk as to what from my perspective, what some of the key issues are and some of the interesting research frontiers. One of the enabling factors in making connectomics so important and brain networks in general is the increasing availability of large-scale recording and mapping techniques, all the way from molecular interactions among proteins and other molecules in cells, all the way up to social interactions in human behavior. In the middle of this diagram sits this very large space of connectomics, which is engaged with mapping anatomical connections, but also functional connections, dynamic connections among usually large, extensive neural ensembles or entire nervous systems. And it is that space that will take up most of my discussion today. So first, I want to say a few words about what makes connectomes special, what kind of architectural features we've discovered over the years, and then turn over to issues of function and dynamics, communication, and ultimately the issue of information, which is very much on my mind these days. One major dichotomy that we need to be aware of in this field of connectomics is that between structure and function. Structural and functional networks have different origins, different ways of being measured and mapped. and some of the methodology that we use to analyze them and model them differ between these two domains. When I first got started in neuroscience, then our knowledge of the human brain was largely confined to a histological dissection, post-mortem dissection of human brains, and then mapping of what you see here, these extensive white matter pathways linking distant cortical regions to each other. Only fairly recently, about 20 years ago, did we arrive at a methodology called diffusion imaging and tractography that allows us to map cortical anatomy, subcortical anatomy as well, in general neuroanatomy, non-invasively in large cohorts of humans. This has given us an entirely new approach to studying brain networks in live human beings, and allowing us to map individual differences and also relate individual differences to behavioral features. We early on engaged with this technique, used it to create a first map of the human connectome in the mid-2000s. And since then, the domain of connectomics has grown tremendously, including resting state fMRI, a new tool to map spontaneous ongoing activity in the human brain. And both diffusion MRI and resting state fMRI have been used to create networks. These networks have different attributes. Structural networks are typically sparse. They refer to anatomic connections, physical wiring between neural elements, whether they are neurons or brain regions, while functional networks are quite different. They are estimates of usually statistical dependency between often remote neural elements in the brain, cross correlations quite often in our fMRI world. So very simple assessments of statistical dependence. They can be much more sophisticated as well, using directed estimates of directed influences or causal influences and so forth. Importantly, though, these two domains of structural and functional networks are quite distinct from each other, and we need to distinguish them in terms of our analyses and our interpretation. When we first mapped the human connectome almost 20 years ago now, several features stood out to us. Individual brain regions had unique connectivity fingerprints, unique sets of inputs and outputs, which presumably were important for their functionality. Overall, the network had a broad or heavy-tailed degree distribution, with some regions and some elements having a lot more connections than others. There were aspects of high clustering and short path length, which are key features of small world connectivity. Very prominent feature was the existence of modules, densely connected clusters of brain regions that are interlinked by hub regions. And finally, the interconnections among hubs, which were statistically more enriched than expected by chance, and formed structural cores or clubs. All these features, which we gleaned on a very small sample almost 20 years ago, have held up very well, I think, in many applications and follow-up studies across many cohorts, and now stand as sort of the hallmarks of how human connectome networks are organized. What about model organisms, though? Because we can do, I think, much more high-quality mapping in using invasive techniques such as histology or even electron microscopic reconstruction in those organisms. What about those networks? Do they look similar to what we see in humans? And by and large, they do. And I'm providing here as a backdrop examples from the fly, the Drosophila nervous system, the non-human primate, as well as the rodent, mouse, and rat. All of these maps that you see here were arrived at with very different technological approaches, from literature collation all the way to track tracing and histology, as well as microscopic reconstruction. And yet, despite these differences in technique, these differences in nervous systems, these networks have similar attributes. They all contain modules. They all contain highly connected regions. They all connect. These highly connected regions tend to be interconnected amongst each other into clubs and cores. So a very uniform, almost we might call it evolutionarily preserved set of motifs in brain networks that seem to be prevalent across different species. Just to flag a recent advance, a really amazing piece of work that was published earlier this month, the full-up reconstruction with electron microscopy of over 100,000 neurons, 130,000 neurons into the software brain of an adult fly with over 50 million connections mapped out in detail. An extremely rich data set published in Nature in a series of papers. And it tells and shows us how far connectomics has really come from those very simple beginnings. I could call them simple because I was part of it 20, 30 years ago. These very small networks that we built back then to these now really quite large and detailed assessments of neural architecture. And just as an aside, it's kind of interesting that AI tools actually were instrumental in enabling the reconstruction of this extremely data-intensive problem of reconstructing in 3 dimensions from electron microscopic images. So AI actually, a very sort of postmodern twist, AI was engaged in mapping connectome networks. And so there's a connection between AI and connectomics, if you wish, that we might discuss later on. Going back to my own work, though, working with Larry Swanson and Joel Hahn at USC, we have engaged in a series of investigations of different subdivisions of the rat brain, where connectivity was derived from published literature that was mined and collated into connection matrices for all these different parts of the rat brain that you see here, from the cerebral cortex, this is where we started, all the way to subcortical regions, now the rhombic brain, and also even the spinal cord. We now have a full map of the brain of the rat, which has 790 regions, 72,000 connections, which have been mapped to directed connections using histological techniques. Connections range over 5 orders of magnitude in terms of strength, from very strong to very weak. The overall brain is about 12% connected, so relatively sparse. We recently published a detailed assessment of the connectivity features of the rat brain. Here's just one highlight of it, and I want to flag the prevalence of modules, because I'll come back to that topic throughout my talk. Modular architecture is very conspicuous in connectome networks, certainly in the anatomy, but also, as we'll see later, in functional connectivity. What we're seeing here is a top-down view of what we call a multi-scale analysis of modularity, where a modularity maximization algorithm was deployed on multiple spatial scales. And the results of that modularity algorithm, which are partitions of the network into modules, were then collated into a so-called co-classification matrix, where each entry of the matrix for each pair of nodes essentially tells us how likely it is to see that pair of nodes as part of a module at any given scale. This is a nice, well-behaved metric between 0 and 1, which tells us something about the propensity of 2 nodes to be found in the same module within that architecture. The multi-scale aspect of modularity is also very important. Modules exist not just at one scale in a sort of trivial manner, but can be found at different levels of scale and detail, and are nested within each other. That is another feature that we find in really almost any connectome architecture we have looked at, whether it's the rat, the human, or an invertebrate brain. There seems to be something about modules that is important for connectome organization. What actually drives the physical layout of connections in a given nervous system? And here I go back to a long-standing collaboration with my colleague Ed Bullmore at Cambridge University. And we speculated, if you wish, or hypothesized some years ago that connectome networks and anatomical connectome networks had complex topology consisting of a mixture of modular architecture and highly connected hubs, and that this architecture was due to as it was expressing kind of a trade-off between two opposing trends. On the one side, a drive towards lowering the cost of connectivity, making connections short and inexpensive, something that is evolutionarily important because brains are very energy hungry and there's a limited amount of space available to actually fit them within a given body plan. So a drive towards economy that would, in the limits, in the limit case, give rise to a brain that you see here on the left. And that's clearly a brain that's not very good at passing information around. On the other hand, a random topology, which is very wasteful to physically wire, would actually be quite good at moving information at fairly fast and efficient speeds from one end of the brain to the other. The actual topology that we find is sort of a mixture. Modularity promotes economy because most connections are made locally within a neighborhood, while the existence of hubs and interconnected hubs promotes the efficient transmission of information. So that trade-off we hypothesized back then is one of the important factors that perhaps drives the existing connectome topologies that we actually see in nature. One very interesting project we were recently engaged with collaborators Yanni Fassab and Yossi Yovel involved the mapping and comparison of connectome networks from a very wide range of mammalian species. These species became available as brains were collected from animals that had passed from natural causes in zoological gardens and in the wild, and then imaged, and allowed us to compare, in a uniformly imaged setting, the brains of species as diverse as carnivores or rodents or bats, all of them mammals. And we ask questions like, as brains change, for example, change size and volume as they do in the mammalian order, how does connectivity vary? And that research project gave us a few interesting insights, which I'll now share with you as to what might be some factors behind connectome architecture in a comparative setting. The first study was carried out by my former grad student, Josh Faskowitz, who got a hold of these over 100 mammalian species, data sets from those species, and asked a very simple question. If we actually compare more closely related, phylogenetically closer species to each other, do we get connectome architectures that are more similar? That is, in some ways, a validation test. We would expect to see that, and indeed we do. This is an important data point because it shows us that our connectome mapping technique perhaps was tapping into real anatomical features in those animals' brains. Going beyond that, though, one thing that we noticed right off the bat is that mammalian brains differ tremendously in terms of their volume and size. In fact, it's 3 orders of magnitude at least. What you're seeing here is a comparison of, first of all, the range of brain volumes illustrated with different subgroupings of mammals, from the bats, which are very small, like Chiroptera, all the way up to primates and carnivores, which are much larger. Then we asked the following question: if we build a generative model that actually tries to statistically reproduce the topologies of these brains using a very simple, relatively simple statistical formula that, for example, incorporates spatial distance and assigns connections based on an exponential decay of density with spatial separation of two regions, what do we get? And what kind of parameter do we have to set our model to for given brains of different sizes to get accurate reproductions of the actual empirical pattern? In this diagram in panel B, what you're seeing is there's a spatial parameter, which is an exponential distance penalty, a negative parameter, because with increasing distance, connection density falls off quite dramatically. That penalty increases as brains get larger, which tells us that in larger brains, as physical distance between neural elements increases, the penalty incurred or the cost of wiring that is incurred by actually creating a physical wire between those separated regions is dramatically increasing. Put it yet differently, larger brains are quite difficult to wire up unless you really pay close attention to spatial separation. Space reigns supreme, if you wish, in governing connectome architecture. The larger the brain, the more important it becomes. A second aspect of this was pursued by my former postdoc, Maria Grazia Puchedu. And here I'm just going to steer you to panel B. We wanted to systematically investigate how modularity differs across different mammalian species in this sample of 103 species that we had available. Once again, brain volume is the key parameter that we're keeping track of. And what this diagram in panel B shows you is that it actually plots a correlation coefficient between the propensity of two nodes to be in the same module, which is our co-classification coefficient, or CC, and their physical distance from each other, which is the Euclidean distance in three-dimensional space. That relationship, which is expected to be negative, the closer two things are in space, the smaller the distance they have from each other, the larger the propensity to be in the same module. So that's a negative relationship. For larger brains, for brains of larger volumes, that relationship is much stronger than it is for brains of smaller volume. To put this in plain English, modular architecture is much more faithful, if you wish, or replicates much more faithfully physical and geometric aspects of the brain as brains get larger. Another way of saying that as brains get bigger, modules become more compact, more spatially confined, and spatially well-defined. So modularity is present in all of these species. And as we go to larger and larger brains, these modules become more and more geometrically confined. Here's a schematic coming from this study, essentially summarizing a few of those trends I just mentioned. Modules in larger brains are more spatially separate from each other, while in smaller brains, they appear to be more intermingled and perhaps, if you wish, more random looking. Modularity is one of the key features of connectomic architecture, and I'll have a few words to say at the end about why or why not AI architectures might benefit from such a feature as well. Turning over to dynamics, so far I've only talked about physical wiring and features of connectomics that relate to synaptic connectivity and white matter pathways and so forth. But now let me turn to the issue of function. One of the questions that we've worked on now for almost 20 years in the lab is how do we get from something that is fairly static and sparsely connected, such as the anatomy of the human brain on the left, how do we get from that to something which is dynamic, always changing, adapting, plastic, as the dynamic recording that you see of human brain activity during the resting state on the right. What is the relationship between these two? How is that swirling mess of activity that you see unfolding in that little movie, how is that constrained, shaped, informed by the underlying anatomical skeleton? We in cognitive neuroscience have, of course, benefited greatly over the last 15, 20 years from the so-called resting state, which is a way of taking a movie like what you just saw, cutting it up into individual frames, and then computing these frames into what we call a simple correlation matrix or functional connectivity. And despite the fact that the resting state is cognitively unconstrained, no task is given to the individual, the results from it, the pattern of correlations that we see, the coherent modules that you see as these blocks in the main diagonal that signify regions that are coherently active over time, those blocks of regions and those patterns are quite reproducible across individuals as well as across cohorts. And that is a remarkable fact given how unconstrained resting state really is. The modules or blocks across the main diagonal of this matrix that you see here have names that we are all familiar with: default mode, default mode network, attention networks, limbic networks, saliency, smarter motor, visual, etc. These have become lingua franca in cognitive neuroscience, and hundreds, if not thousands, of papers have been written about the functionalities of these systems in not only resting state, but also in task. I will leave all that aside. Just mention at the bottom of this slide one more time that resting state is quite reproducible, and that actually gives me at least a hint as to where it might be coming from. The main message is that a lot of the variance observed during resting state actually is explainable on the basis of the underlying anatomy. One of the features of resting state that was initially underappreciated, but is appreciated more and more these days, is that functional connectivity in resting state itself is dynamic. It's not a static pattern. In fact, the whole name resting state is a misnomer in some ways. It's not a state, and the brain is not really resting. It's constantly moving. And we know this from theoretical proposals and investigations that suggest that there's a constant ebb and flow between a more segregated state of the brain, broken into coherent modules, versus a more integrated state, where these modules become interactive and interlinked. We see this empirically in resting state, and as found by studies carried out by Andrew Zaleski and Michael Breakspear many years ago now, distinguishing a more low efficiency versus a high efficiency state during resting state, and that is their way of expressing functional segregation and integration. Work carried out by Max Schein and Russ Poldrack, a similar finding here, an alteration during resting state between a brain moving from a more segregated pattern into a more integrated pattern, and vice versa. So this ongoing dialogue between modules and their integration appears to be a baseline feature of how brain dynamics unfolds. In our own work, we've been trying to track this initially with what everybody else was doing, which was windowed analysis of resting state time series. Windowing has I would say unwanted or suboptimal features associated with it. We were looking for a better way of doing this, and we found the following approach. I should say Rick Betzel and my student Josh Vaskowitz found this approach. Here's how it goes. We're looking at nodal time series. These are activation time series coming from, let's say, a single region of recorded, let's say, during resting state. These are normalized time traces, Z-scored. Two of them. What we typically do is we simply multiply element by element, moment by moment, these two contributions from these two time traces together, which gives us what we call an edge time series. This informs us about the co-fluctuation state of the relationship between these two nodes. Are they co-fluctuating in the same direction, in which case we get a positive signal, or are they co-fluctuating in an opposing direction, in which case we get a negative signal? Interestingly, this edge time series, when we take the global mean of it, is exactly equal to our Pearson correlation, and is therefore strongly related to the metric of functional connectivity that is very uniformly employed across many studies in fMRI these days. So it's actually a very natural way of decomposing a functional connectivity estimate, previously delivered only as a single number affixed to a long recording, decomposing that into moment-by-moment contributions along the way. When we do that, we get these edge by time matrices, which have, as you notice right away, an intriguing sort of vertical organization to it. I'll come back to that. When we take this edge by time matrix and we take the mean across the x-axis, we're getting a vector, which we then can refold into a square matrix, and that is our functional connectivity. So nothing has been added or taken away from the classic approach, except we have unfolded functional connectivity into moment-by-moment contributions. When we look at these vertical features in this edge by time matrix, they correspond to highly moments of high co-fluctuation that we called early on in the game events, perhaps slightly the term event is a little bit loaded, I might say, but they were conspicuous in this edge by time matrix. So these are moments when there is high co-fluctuation, either high agreement or disagreement among all or many of the nodes in a given brain network that we're recording from. Turns out these moments occur in virtually every recording we've looked at from any human being, as well as in animal recordings as well. So they are not artifactual. They are not obviously related to potential artifacts in fMRI analysis, such as motion or other physiological processes that are unwanted. So they appear to be a product or a byproduct of regular neural activity. The edge, there's a whole edge-centric approach building on edge by time or edge time series analysis that I do not have time to go into. We have and others have pounded on that for several years now with a lot of interesting applications, all the way from fingerprinting to brain behavior relationships to movie data and so forth. So there's a lot that's come out of this approach in the last few years, and I don't have much time to go into detail, unfortunately. One thing I wanted to show you, though, is a movie, just to drive home the fact that functional connectivity is highly dynamic, and that the features in functional connectivity that we are so fond of in our field, such as those networks that I mentioned earlier, default mode, frontoparietal, etc., are really quite fleeting on a moment-by-moment basis, are actually not strongly expressed. I'll start this movie, and what you see is I'm populating my edge-by-time matrix as my recording proceeds. At the bottom, you see our summary statistic of co-fluctuation magnitude. There was a very strong event happening just now that you saw in this peak. If you guide your attention to the movie frame at the top middle, that plots the co-fluctuation magnitude for every TR or every frame of the fMRI recording. If we take the global mean of the movie that you are seeing here, we're getting our familiar functional connectivity shown in the upper left. The point I want to make is those blocks that you see on the main diagonal, those are the systems that I mentioned earlier, those coherent, quite reproducible features of functional connectivity, such as default mode, visual network, attention networks, and so forth. And I want you to notice that at any moment in time, those systems are not necessarily strongly co-fluctuating. While we get on average a picture such as the one shown on the left, quite reproducible, at any moment in time, the co-fluctuation state of the system we are looking at, the brain, is quite variable. It's not as well organized into these cohesive domains. And there's a lot of structure that I think we can now tap into with this decomposition technique. That we have done so, as I mentioned, in a number of follow-up studies that have come out of this work since then. Just one last point about this. Once again, I want to underscore that the systems that we're so familiar with in cognitive neuroscience, that many of us are writing papers about, are expressed intermittently. That is, they come and go as we are recording a resting state scan, let's say. And this, to me, is still an underappreciated fact about resting state fMRI. The fact that those systems that manifest very reliably when we look at longer averages of recordings are actually not present moment to moment, are statistical features, really. There's a lot that we've done in our lab and other labs have done as well, following up on this work. And I have no chance of even scratching the surface in this very brief talk. So I would have to direct you to those other papers, or perhaps we can talk about some of this in discussion. One thing that we do a lot in the lab and others do around the world as well is doing computational models and simulations of brain networks, taking an anatomical pattern and actually using a connectome architecture as an underlying coupling structure to, for example, couple oscillators to each other and get dynamics from that. We have done that over the years. Others have done so for sure. One thing that we found interesting coming out of this work on dynamic functional connectivity, we wanted to see, do we see these events in neural simulations? The reason that might be interesting is because in neural simulations, we can investigate where they come from in a more invasive fashion because we have all our parameters in front of us. And we can also reasonably exclude physiological artifacts. So this is the work of my grad student, Maria Pope, who has coupled up a classic model of coupled oscillators, the Komodo oscillator with a phase delay with phase delay coupling to create reasonably realistic dynamics that can be treated as if they are synthetic fMRI recordings. What we see is that we find, without making any adjustments in parameters, in fact, there's hardly any parameters in this basic equation of the Komodo oscillator, we are getting event-like excursions of co-fluctuations very readily, and at about the same frequency that we see in empirical recordings. And in her work, Maria actually traced these events to synchronization patterns that occur within modules in the underlying connectome network. So once again, modules are critical for actually bringing about these fluctuations that we see so prominently in empirical recordings. When we destroy the modular architecture, dilute the modules, or essentially decompose them, create a random network, as it were, we do not get these excursions. And we can trace them in the model very faithfully down to synchronization events that are really module specific. So these are not just statistical features in fMRI recordings, in my opinion, but they reflect a particular mechanistic origin of high co-fluctuations, which are very important for shaping functional connectivity, as it turns out, tracing them to connectome architecture underneath. I want to turn briefly to the issue of communication and communication models, which are an important topic in network science. We have applied communication models to connectome architectures, especially most recently my former postdoc, Kaio Seguin. Here's the problem that we're tackling with that approach. A structural brain network is sparsely connected. If two regions or two nodes wish to communicate or exchange a signal, there's a difficult problem, which is what path should that signal take? This is a non-trivial problem for which in mathematics or computer science, we have fancy algorithms to address the problem. But in the brain, it's not obvious how the brain might actually solve this issue of discovering a path between two nodes that are perhaps wanting to exchange signals. Most of these paths are going to be multi-step and not going to be direct connections because the brain is sparsely connected. And the number of available paths, as many of you know, if you're doing a little bit of combinatorics on even a small network, is truly astronomical. So the problem is non-trivial. So the approach was to take structural connectivity patterns, connectome matrices, implement different communication strategies on these matrices, different models according to which communication might take place, and then create from that a so-called communication matrix, a matrix that defines for each pair of nodes how easy or how hard it is to communicate between those two nodes, given the policy, given the network communication model that's been implemented on the connectome. This communication matrix can then be compared to empirical recordings of, for example, functional connectivity, which presumably gives us an estimate of how much communication occurs between a pair of nodes. So one study that Kaio has carried out, before I go there, I go back to my yet former postdoc, Joaquin Goni, now professor at Purdue, who actually really blazed the trail for the work that Kaio did by implementing different network models of communication, finding that it was a very interesting strategy called search information that was very highly predictive of empirical functional connectivity in the human brain, and which gave us confidence that we could in fact pursue the strategy of implementing simple communication models on graphs and get a reasonable estimate of predicted functional connectivity. Here you see a comparison in this diagram of an empirical recording, functional connectivity on the left, and then a predicted pattern of functional connectivity based on a communication model on the right. As you can see, a very strong level of statistical overlap between these two empirical and modeled approaches. That's, I would say, a non-trivial outcome. There's no reason why we might a priori believe that a human brain might obey a particular communication policy. And yet, several of these policies, such information among them, are actually quite successful in modeling communication strategies. Here's where I wanted to come to Kaio's recent paper in Neuron a couple of years ago now, in which he did the following, using a large empirical data set coming from the French lab of David et al., which comprised recordings, multi-electrode recordings in epilepsy patients that had been implanted with electrodes as part of pre-surgical testing. That data set contained literally hundreds of thousands of stimulation experiments in which a current was delivered to one electrode, and then the ripples of that current going presumably through the connectome architecture, the echo of that perturbation in terms of other sites responding across the brain was being recorded. Given that we think we know what the connectome looks like, given that we know where the stimulation electrode was located, we can then reconstruct potential paths that lead us from a stimulation site to a recording site. We know from stimulation and recording pairs which stimuli were particularly successful in eliciting a response. We can then implement a communication model such as shortest path efficiency, SPE, search information, SI, or communicability, which is based on diffusion. And we can compare for these hundreds of thousands of stimulation experiments, which of those policies is most successful in replicating the empirical finding. We're comparing, in other words, the ability of a given communication policy to predict the pattern of stimulus and response that we see in the empirical recordings. It turns out that the search information is one of the most successful ones. Diffusion-based communicability is close behind, while the shortest path, a routing-based strategy, is the least successful. While this will not allow us to directly pin down which communication policy might be implemented in the brain, we need more data for that, it does provide a framework for evaluating different ways in which network nodes might be communicate in a nervous system with each other. In a review article, Kaio very nicely contrasted different strategies of communication, and there's one spectrum that we pay particular attention to. On the one side, random walks, which is essentially diffusion, very wasteful in some ways, message broadcasting indiscriminately in all directions out from a given stimulation node. On the other side, shortest paths, the most efficient way to communicate, but also the way that allows, that requires the most information about the network topology. Finding a shortest path in a network is a computationally hard task, and it is not obvious to us how a brain network might implement such an algorithm. So there's a spectrum of possibilities from wasteful diffusion to efficient shortest path routing, where in our own work, the most successful algorithms, communication policies, appear to be more on the left side towards diffusion in terms of predicting functional connectivity. So the picture up until now looks about like this. We have on the bottom end, on the foundation, the connectome, a structural and anatomical wiring diagram, what's connected to what by virtue of synaptic connections and axonal pathways. On top of that, functional connectivity, often recorded from whole brain imaging or multi-electrode recordings invasively, and reporting out essentially a statistical dependence, an alignment, sometimes linear, like in Pearson correlation, between the activities of observed neural elements. Notice that functional connectivity is in some ways non-mechanistic. It makes no statement about the underlying mechanistic process. It is non-causal. It's typically undirected. But it does emerge from signaling events that occur within the connectome. Those signaling events themselves set up a very highly dynamic underlying communication network, which consists of directed flow of information from neural element to neural element along many directed pathways. It is this middle domain of communication dynamics about which I believe we know the least. We have fairly good access to functional connectivity. We have fairly good access now to the connectome architecture on the left, but we are lacking good ways of empirically following the blow-by-blow pattern of communication that occurs in the underlying structural network. A few words about information and the key role that it plays in putting together, in some ways, networks and dynamics. I only have a few minutes left, so I'm going to be very quick on this. Perhaps we have time to discuss this issue more. Again, this goes back to work that carried out by my former graduate student Thomas Varley, now postdoc at Vermont, and current grad student Maria Pope. Just a few words about this, just to whet your appetite, perhaps. When we look at neural information, especially multivariate neural information, there's sort of two different ways or two different modes in which multivariate information might be organized. One is perhaps simple to understand, and that's redundancy, simply replicating information across many neural elements where the information itself is kind of identical and just simply copied. A much less well understood and much less well researched aspect of multivariate information is what we might call synergy, which is the integration of unique information into a whole and a pattern that forms an integrated whole that is not trivially separable into individual components, where the whole emerges from the interaction. Once you take the interaction away, there's nothing left. So synergy as integration of unique information coming from different sources. My favorite analog here is that of a string section in an orchestra, where all the strings play the same set of notes. That's redundancy. Versus a jazz ensemble or a string quartet, where every one of the voices is unique and where the music really emerges holistically from the interaction of the individual players. If the players were playing by themselves, it would make no sense at all. It's the interaction that gives it that holistic aspect that we enjoy. There's math underneath all of this, which I will not get into. In my case, some of the mathematical frameworks go back literally 30 years to my work with colleagues Tononi and Edelman at the time, configuring something that has become known as TSC complexity. I'm the S in the middle, that quantifies over multiple levels of scale in a given system that we observe how information is distributed. And there are interesting modern formulations that play into this TSE formulation as well, such as total correlation and dual total correlation, and the difference between the two, which we call O information, which is a proxy for identifying synergistically or redundantly dominated subsets in a system. Looking at the balance between synergy and redundancy in brain recordings, one of the questions that Maria and Thomas asked early on was, can we actually identify parts of the brain for which synergy is more dominant than redundancy in the dynamic evolution of those sets of nodes? And the answer is yes. And we find synergistic subsystems predominantly actually in empirical fMRI recordings involving the regions that you see here in the middle panel at the bottom. And they do not line up very well with those functional systems that we talked about earlier, like default mode or frontal-parietal or visual. Instead, we find synergy, and perhaps this is understandable given the definition of it, not within a given functional system, but between these systems, in the interstices, in the intersection of these functional systems. And that is documented using some statistical measures of where negative O information, that is synergistic subsets, actually live. They are predominantly found in the intersection of multiple functional systems in resting state fMRI. Just to give you a bit of a big picture and then a few thoughts about AI, to which I want to return at the end. I've talked about networks a lot. Network science approaches have been really influential in neuroscience recently, from connectomics all the way to molecular genetic social networks. I've mentioned the importance of modularity and the issues of cost and balancing economy and efficiency. Dynamics is obviously equally important, and there are some buzzwords here that I want to mention: synchrony, dimension induction frameworks, flows and manifolds. These are all aspects of neural organizations that people are investigating intensively. Networks and dynamics have an interesting interplay with each other. I mean, networks constrain dynamics. Dynamics can connect back onto networks to plasticity. So there's interesting aspects in the intersection, temporal networks, coevolution of dynamics and topology, for example, in plasticity. Emerging from this dialogue between networks and dynamics is that ever important domain of information. Even though I devoted only 5 minutes to it, it really looms large in my own thinking. In some ways, information, generating complex information, synergy perhaps, is one of the key aspects of brain function that we should be paying attention to. So there's complexity. That's one of the emerging features. Higher order interactions, and a very active topic in networks and network neuroscience these days, and the spectrum going from redundancy to synergy, which has been underappreciated so far. All of this kind of forming a sort of a triangular intersection or interaction. And to me, these three ingredients, networks, dynamics, and information, are the three key things that we need to understand theoretically, computationally, to really understand how connectome networks operate and how the brain really functions. Now, just a few words about the connectome and AI. Sometimes it seems to me, the connectome is like a bird. It's a natural, it's a structure we find in nature that does interesting things, while AI is like an airplane. It's engineered, it's powerful. It is what we use when we want to go from one continent to another. I wouldn't take a goldfinch. And it uses some of the same underlying physical laws or physical principles, but it is quite different from the capacities of a natural system such as a bird or an actual connectome network. So there's some parallels. Yes, connectomes and AI architectures feature nodes and edges. Obviously, this is a major parallel. Distributed processing and representation, and of course, plasticity and learning are present in both. But I think there's an ever larger number of divergences, areas of contrast between the two domains. And I don't know if these are irreconcilable contrasts, or if they are contrasts that perhaps will be bridged or overcome in the coming years. So for one thing, connectomes are recurrently connected. There's not really a multi-layer architecture with a mainly feed-forward flow of information. It's recurrent. Sensory regions receive most of their inputs from the inside of the brain, not from the sensory surfaces to which they are connected. Connectome networks are invariably sparse. In the case of the recent Drosophila connectome, the connection density was something like, I think it was 1/10 or 100th of a percent. Extremely sparse architecture. modules and hubs are prevalent in connectome architectures. Once again, the software brain contains highly connected neurons as well. Brain regions in the human brain are not all alike, but some are more densely connected than others. And there's modular architecture across multiple scales. Connectome networks are by necessity built and operated in an economic fashion. They must conserve natural resources such as energy and space. There's no way around that. Evolution does not get to overspend on what it is given. That's the road to extinction. So there's a strong emphasis on economy in connectome architecture, something which is not the case, I think, in AI systems currently, as they currently exist. There's complex dynamics, nonlinear dynamics that is actually happening in connectomes. I've shown you a few examples of that, perhaps not as prevalent in AI architectures as well. And connectomes, the brain, nervous systems are active even when there's no sensory input present. This is the resting state that I mentioned earlier. Even when there's no obvious task to be accomplished in the environment, nervous systems tend to be active. This plays into their generative or predictive function, as opposed to being just reflexive and waiting for inputs and generating outputs on demand. Of course, networks like the connectome are embodied. I have a whole other career that I abandoned about 20 years ago in robotics, which taught me a lot about the importance of interactions between neural networks and the environment within which they find themselves and how they interact with the environment through physical bodily movements, shaping the environment, also shaping their own inputs, generating inputs along the way, not just waiting passively for inputs to arrive. Finally, connectomes are materially in flux. They are living things. The carbon atoms in your body turn over in about a week. This is true for the connectome as well. It's constantly rebuilt again and rebuilt again. It's like an airplane that actually is changing out its material substrate as it's aloft. And finally, learning, and my colleagues in developmental psychology can speak much more cogently to that. Learning is quite efficient in organisms. Babies don't need to be exposed to a million faces to understand what a face is. And that is something that, again, is a point of departure between connectomes and AI. So I've covered all these different aspects. I really look forward to the discussion. I wonder what you make out of all this. And I would be remiss if I did not mention that in my spare time, I'm operating a journal that I founded some 10 years ago now in the field of network neuroscience, which is a growing area in neuroscience. Network applications are constantly on the rise. Connectomics is just one aspect of that. So with that, I thank you for your attention, and I'm curious as to what you make of all this. Thank you.
Thank you very much, Olaf. We already have some questions. I want to use the chairman's prerogative to ask the first question,
Sure.
though. If one's primary interest in connectomics is cognitive rather than general biological, have you, for example, compared something that's undeniably cognitive, which is learning, before and after learning something, changes either in structural or functional connectivity? Yeah,
it's a great question. I have not personally, and I'm much less. I've been, because of the prevalence in some ways of all the issues surrounding connectome mapping itself and resting state dynamics on top of that. I have not done a lot of work on learning myself, but it is, I think, well recognized in the field empirically that functional architectures, even resting state networks, which are presumably relatively stable over time, do change with exposure to stimuli, with practice, with learning routines, and so forth. So there's a lot of sensitivity there to ongoing activity. The ongoing activity does inscribe itself on the weights, presumably on the weights of connections, which then in turn change the way that these dynamics unfold. So that dialogue between physical change in the underlying anatomy and plasticity therein, and dynamics, which then also changes as a result of that and inscribes itself on the anatomy, that is something that people do indeed pay attention to.
Thank you very much. And now the first question is from Jean-Pierre Briot.
Yes, thank you, Olaf. A great talk and a great series of seminars, Stephen, also. My question is about the issue of control of the dynamicity of the networks. That is this control at the same level of the modules? That is, one module can in a way trigger reconnectivity, or is there some kind of higher level, different level in a way, modules which are control modules, specific, which to make this reconnection? Just quickly, I'm on computer science, and I'm interested in the creativity processes, being artificial or human. And I read recently some interesting study about jazz improvisation process, where there's the default mode network involved for, in a way, creating new, motifs, melodic, or etc. The saliency network, in a way, tagging it as potentially interesting, and apparently making the kind of switch of for the activating the executive control in order, in a way, to build up on the skeleton of melody, etc. So so I'm interested in your
I mean, in some ways you answered your own question by referring to what I was going to say, namely that there are elements of the brain, including the networks that you've mentioned, that are clearly engaged in, if you wish, directing the flow of interactions in the more basal or foundational modules that we have available to us. So that is one way that people think about this. The frontoparietal network or the default mode network as being sort of supervening entities that are directing and channeling information flow. Having said that, it's not clear mechanistically to me how that might operate in a very distributed, extremely complex architecture, such as the human brain, which is devoid of any sort of top-down tracking, or there's no knowledge anywhere in the system, as far as we know, of its own topology, or of how it's laid out. There's no map engraved in the brain itself that the brain can make reference to. It just does what it does, and it's in that sense a dynamic system. And the trick, I think, is to find from the bottom up, using just the tools of dynamics and networks, find mechanisms that can actually operate at this more supervening and longer time scale, that actually can inform us as to how the brain finds a trajectory, through this extremely high dimensional space of going from state to state to state, not randomly, but in a way that actually makes sense in the context of behavior. And that's not an answer to your question. It's more like a reformulation of the problem is, unfortunately. But I think, the glass is half full. I think we have now a few ingredients available to us and tools and concepts that we can think about. And also, we're still at the very beginning of this endeavor. 20 years ago, I remember the time quite well. We had very little information about human brain networks. In fact, nobody was working in networks at all. So we've only gotten a very small a snapshot of what networks look like and how they interact with dynamics. But the question you're referring to is actually very much on my own mind. I teach a course on the subject to undergraduates. I have a class this afternoon, it turns out, on networks and cognition, and where we will talk about exactly this issue and review some papers on this. it's very much an open question, but a very, very interesting research area. I think you might agree to actually get traction in.
Thank you, Olaf,
Thank
and
you.
merci,
If I may,
Jean-Pierre.
sorry,
Est-ce que tu as...
if I-
Vas-y, vas-y.
Okay, yeah, thank you. I know I assume that there's no knowledge, I mean, consensual, and it's a deep question. But you know, the issue of control is fundamental because, for instance, as my experience, own experience as when I do meditation, in a way, the default networks just gets my attention without any control, explicit control. He takes my attention in a way. So my point is, but in your knowledge, your experience, etc., did you find some possible kind of modules, networks, which could be more specialized about control, or is it really more emergent, in a way, a competition for attention and for connection between the modules so far with current knowledge?
My intuition is it's a bit of both. So I think the modularity that you refer to, the fact that there's modules in the first place, what does that really do? It serves to reduce dimensionality of the system, right? I mean, modules are almost like sort of principal components. They are ways in which we channel the otherwise very large manifold of where the brain can be into a much smaller space. There's a constraint, you know, and then there's a language on top of that where I think modules get dissolved, reformed, combined, which then give rise to the states that you're referring to. Now, is that control in the classic sense of something tracking this and putting it together, or is it bottom-up, emergent? Now, if it was just me, I would say top-down control can emerge from interactions at a very elementary scale, perhaps with the help of modules as ways of building components, components. So think of cognitive architecture as a way of combining like a Lego set, you know, a set of preformed things which are not random themselves, but kind of engraved in the Connectome architecture. And now there's a way to combine them in a way that is still very large, a very large space, but not infinite, okay, and unmanageable. So that's kind of where what I'm thinking.
Thanks a lot. Thanks a lot.
Thank
Very
you.
interesting. Thank you.
Now, I have here a bank of a priori questions based on the references that you had, and some of them were answered by your talk. But there's one subset that might want to express itself here. Iris, do I put you in a spot? I like your edge-centric question. Do you want to ask it, or shall I ask it for you?
All right. Let me put it.
Can you hear Iris?
Yes.
Okay, go ahead.
Right. Just one second, please. I'm sorry. Yeah. Okay. So my question was about neuroplasticity. And more specifically, how can the edge-centric perspective represent neuroplasticity? And can connections be remodeled? Yes. And how can it be represented and analyzed through the edge-centric model?
Yeah. So I think you know, in the work I've presented, plasticity did not feature very prominently, as you may have noticed. You know, I was referring to dynamics and dynamic states that change over time without really making reference to underlying changes in anatomy or synaptic strength or modification thereof. So the answer is probably, you know, in principle, the edge-centric approach is very, very, very general, right? I mean, we have been operating in network science for the most part with a node-centric approach so far. We've taken the nodes as being our elements, and then interactions were sort of secondary to that. You can flip the script and take the interactions as being the primary component, right? And that's where edge time series come in. Edge time series track all pairwise interactions in a given system, all of them, not just the ones on anatomic connections, but also the ones that are not supported by a direct anatomical link. And of course, if there is plasticity in the system, that will transfer to not just changes in node activations, nodal activations, but also changes in the edges. So I think in principle, absolutely yes. But for reasons of the sheer richness of what we've been working with in the past, we have not made a step yet into real plasticity or learning using the edge-centric approach. I know others have tried. In principle, it can be done, yes. And I think the edge-centric approach might be a more natural way of looking at plasticity, because after all, it is the connections that are plastic. And edge-centric approaches should allow us to actually track this in much finer detail, if that makes sense.
There was another question about edge-centric approaches. Fonctionnaire, est-ce que tu voulais poser ta question aussi?
Thank you for the conference. I would like to know, how do you see the evolution of the use of computer models and connectomics to map and predict functional dynamics of the brain, particularly with regard to understanding neurodegenerative diseases?
Yeah. So there's two parts to your question. First of all, the computational models that I've mentioned in just one instance to you, the core motor oscillator model, that's a whole field in itself, and I think a very productive one, and one that I think will be taking advantage of Connectome data sets in the future for sure. So even in the recent publication of the Drosophila data, which were extremely voluminous, people immediately, even in that set of papers, immediately implemented Connectome data in computational models and tried to make steps towards predicting functional connectivity, for example, or effective connectivity from the underlying synaptic architecture. So that is an activity that I think would be very productive. And I think it is productive because in a computational setting, we are forced to adopt mechanism, right? We have to write down equations that tell us how we're going to do this. And so computational models are explicit, like instantiations of hypotheses about how structure translates to function. So that's very productive. You mentioned degenerative conditions. This is very dear to my heart because I have collaborations with people who work in dementia, and we are looking at data sets coming from clinical studies quite regularly, including dementia. And here, I think the application of network neuroscience is quite straightforward. I mean, we adopt a network approach. We can track or measure which parts of the network are disabled or degenerating or subject to atrophy or subject to injury. And then we can extrapolate from that onto what functional systems are impaired, and perhaps from that what cognitive capacities are particularly at risk. So the clinical applications of network neuroscience are very front and center in many people's, in many individuals and many groups' work out there, including our own. And I think it's a very productive future application of connectomics in a clinical setting. We need, however, better data. We are currently still somewhat stymied by the relative lack of spatial and temporal resolution in our non-invasive recordings and our lack of understanding of the underlying cellular architecture. So I think there's a lot more to come in the coming years and decades in computational modeling that is applied in a clinical setting.
Thank you, Olaf. Serge Robert seems to have raised his hand and then lowered it. Serge, tu voulais?
Yes, why not? Okay. Well, I work mainly in the field of cognitive science of reasoning. And in one of your conclusions, you say that connectomes are more predictive and generative than reflexive. Do you think that we could find there a neural biological foundation for the prevalence of system one spontaneous reasoning strategies that we use so often, the prevalence of system one reasoning strategies over system two reflective, critical, and analytical strategies of reasoning?
Yeah, I mean, I am not an expert in this area. It's a very good question. I wish I was more more of an expert in cognitive science, to be honest. I meant my remark more in the context of, again, dynamics, right? Reflexive dynamics to me is a brain that stands still and is doing nothing when there's no input. An input comes in and then an output is computed. Sort of the old, input-output mapping that we're so familiar with. While in a biological system, in a biological setting, it does appear that spontaneous activity without an obvious engagement from an external stimulus or any given task that is imposed is quite important. And it wouldn't be there if there was no function for it, because biology wants to be as cheap as possible and not waste energy. So why would we have spontaneous activity if there was no role that it plays? So these are just dynamic arguments, nothing to do with cognition yet. But I think there is probably a very good, very deep reason why spontaneous activity occurs that is ultimately cognitive. And here is where I think future, AI architectures, which currently, as I understand, do not really implement spontaneous activity in the absence of input, might benefit. Because I think where AI is weak is exactly in the domains that you mentioned. It's not self-directed, it's not predictive. It is largely reflexive, in my opinion. I may be wrong, and please, if there's AI experts in the audience, please correct me if I'm wrong, but that's my reading of what AI is really like. And so it's really capturing, it's really very good at one thing, but it's not really not good at something that is actually cognitively much more exciting and interesting. I can't say too much about your specific reference to System 1 and System 2, but I hope that some practitioners in connectomics currently or in the future will be able to make that link. Because, again, in my own work, largely neurobiological, we haven't gotten quite as far yet, but it is an important question that you're raising, and one that I think will have ultimately an answer that is rooted in connectomics.
Thank you. Very exciting.
Yes.
We have several questions. Several people want to ask questions, and one that I'm going to elicit first. So I'm going to first, Hamid, are you there?
Yes.
Are you willing to ask your question? Because you were asking about dynamics as well, or do you consider?
In order to have a effect on higher order
Speak loud.
brain function, is it possible that we can shift emotional state, or if we can change diverse in emotional valence from your study?
Well, so now that's I suspect yes, but again, emotion is something that I have not spoken to today or in my work ever. I don't know quite how to address this.
That's the risk if you say the word dynamics to a cognitive.
Yeah, if you for me, dynamics is more, it's a physics thing, right? It's coupled differential equations and attractors and things like that. So I'm trying to think. I mean, how would you define emotion? And let me ask you that. What there's many definitions, as I understand. What is your favorite framework within which to place that concept?
Perhaps we won't continue in that direction because it's getting remote from the original talk.
Okay. Yeah.
And there's some people that say we're running out of time, and there's some more people who want to ask. It was actually Professor Villemer who was first, and then Pierre Vadnet. So Roger, vas-y.
Okay, thank you very much. Thank you very much for the talk. Maybe another question about the contrast that you made between the Connectome and development and AI. That's something I would like to hear you about, because there's a major there's a major difference in terms of computation, because in the Connectome, everything is happening at the same time. So it's highly parallel. And in some sort of way in computer science, we don't really understand how to make highly parallel algorithms or even machines or things like that. You have multi-cores and stuff like that, but essentially computation is very localized, and even when you have application in AI, you have localized computation that every part of the algorithm is executed. There's many parts at the same time, but not at all at the scale that you have in the Connectome. So would you have any insight about how can we understand highly parallel computation like you have in the Connectome?
That's such an easy question. I laugh because it's a difficult question. So here's a thought that I would offer. Yes, Connectome, or a neural network that operates like an analog computer is highly parallel and decentralized on the surface of it. There's no clock. There's no synchronizing process that all of the elements have to obey. However, when I come back, there is modularity at multiple scales. Now, what modularity will do, it will introduce spatial inhomogeneity and sort of cross-graining the system at different levels of scale, right? So so it's not just a random network which is operating, but it has some major dimensions that are defined by the underlying modularity. And secondly, modularity in dynamics will give rise to timescales that are much longer than the operating of each element, than the operational timescale of each element. So for instance, these events that I pointed out to you that we see in resting state, both empirical and simulated, follow from synchronization patterns that unfold in clustered architectures. And these synchronization patterns, when you plot, for instance, an order parameter or a synchronization parameter over time, that will operate on a time scale that is, orders of magnitude slower than what you see in the individual element. So there's timing and time scales that emerge, for lack of a better word, from the underlying architecture that are perhaps providing a scaffold for interactions to occur in a more organized fashion, if that makes sense. So it's not as parallel as perhaps we make it out to be. There is actually a language to it, but it is a language that comes from the dynamic interactions and from the structure of those interactions, as opposed to from a central algorithm. The algorithm is the architecture, okay? That's it. There is no code, as far as we know, that's running on in our brain. It is, however, there's code ingrained in the way the connections are laid out. That's the way I think about it. It's the sparsity of the connections. That's the code, okay? and that defines that some interactions are more likely and others will never occur. And so I think that is where control ultimately comes from. That is where flow comes from, the ordered progression of states in a sequence. But I'm not. I think we're not fully. I'm waving my hands a little bit because it's difficult to get our arms around these concepts at this stage. But that's where I feel like that's the flavor of what we're aiming at, if that makes sense at all.
Okay, thank you very much.
We're forgiven for waving your hands at this stage of the project. You're congratulated for the enormous progress that you're already making. Now, I think that we're running out of time. I think Pierre Vadnet will probably be the last speaker, unless somebody raises their hand extremely quickly. Go ahead, Pierre.
Thank you. Very good presentation. Gives us lots of info to digest. But to start, I would have two very simple questions. first of all, why do you talk about nodes and edges instead of neurons and synapses? And the second question, and about the same thing, is does the connectome have input and output layers?
Okay, nodes and edges. I adopt the language that is coming from network science. In fact, there's other terms for nodes and edges that I'm not using, like vertices and so forth. And it's simply neurons and synapses refers to a particular physical entity. And neurons and synapses are physical things that we know from neurobiology exist, while nodes and edges is more of a graphical or mathematical concept that can apply to elements of different kinds, including brain regions or neural populations that are actually consisting of hundreds of thousands of neurons. We can still call that a node. So it's a more general framework, and also it relates then to other subsequent constructs such as adjacency matrices or connection matrices, and anything else that we can build from there, including modules. Modules are a network science concept in my talk. And input and output layers in the connectome. Yes and no. I mean, some of the more recent reconstructions, including Drosophila, people have actually tried to design or discover paths that go from sensory neurons to motor neurons, and we might consider sensory and motor neurons as being our input and output layers of the nervous system. And people have been using graph theoretical tools to actually map these map the transformation of sensory inputs, sensory neurons, into motor neuron outputs going through the connectome. But having said that, one of the features of the connectome is that it's highly recurrent. So the flow of information is almost in no case unidirectional. Motor neurons and neurons that are closer to the output side of the brain are also acting back on inputs. This is something that perhaps is that's one of these distinctive features of connectome architecture. It's not a feed-forward network, as it is often implemented in AI applications, but much more recurrent, where the layers are actually bidirectionally connected, if that makes sense. So yes, there are input nodes and output nodes that we can sometimes identify, especially in cellular reconstructions, as sensory and motor neurons. There are paths between them, there's flow of information between them, but the flow is also recurrent. Not sure that answers your questions.
Thank you very much. But what is the simplest connectome that you have identified?
Well, the oldest and one of the smallest connectomes is that of C. elegans, which is a nematode living in the soil. We've had that information, that connectome pattern, since the late 70s, early 80s. It's 300 neurons with about 2,500 or so chemical synapses. So that's a relatively small nervous system. And yet, despite the fact that it's modest in size, the complexity is overwhelming. In fact, there is enormous amount of information available on this nervous system, on the rest of the worm, and on its development. And even though we have had this connectome available to us in its entirety for over 40 years at this point, it has not actually provided us with all the answers about the behavior of this creature, which is another sort of interesting parallel to genomics. When we first coined the term connectome 20 years ago, it was in deliberate analogy to the genome being, the set of all genes. And I remember the day in the late 80s, early 90s, when genomics was first emerging on the biological scene, and people had all sorts of inflated expectations about what genomics would be doing. It would solve all of biology in some people's accounts, and it has not done so. In fact, we have genomics. We've had genomics now for 25 years, and we have a lot of genome architectures that we've mapped, and yet how genetic information is translated into, into biology is still a very complex and challenging task. So I think, building on your question a little bit, even as for a simple nervous system that consists of 300 neurons and 2,500 connections, very modest in size, we still have no way of easily translating that into a complete account of what the organism does or what its behavior looks like. And that should give us pause. Connectomics is fundamental, I think. It is a part of the tool set in neuroscience that we cannot live without anymore, but it will not be the be-all and end-all or the answer to all questions that we might have. The questions that many of the audience members have asked me today are the kinds of questions that remain, and even though we have the Connectome as a tool and as a foundation, perhaps, questions of control, questions of how everything relates to behavior, plasticity, and learning still remain. And now we simply have a better tool, perhaps, to ask our question in a more intelligible fashion. So even though I'm proselytizing about the Connectome all the time, I'm not one of those people that says it's all we need to know about how the brain works. It's actually necessary, I think, but it's not sufficient. That seems like an appropriate remark to end.
We've run out of time, so what I'd like to say is that we're very grateful for this informative and thought-provoking presentation, which was also very forthright about the limitations of what's known so far. I think you still look optimistic. So I guess that means that in the next 10 years, you're expecting more.
Well, I think it's hard to, by definition, it's very hard to tell what the future will bring. And what we tend to do is we just extrapolate linearly, right? So we see where we are today, and then we kind of extrapolate into the future. And that's usually not how it really works. There's non-linearities along the way. And my hope is that we will continue to see technological developments that are, accelerating progress. And also, I hope, conceptual developments, because I do think that theory and cognitive science are playing an important role in helping us understand how the system really works together. I'm not a reductionist. I've never tried to make myself into one of those people that think that we can boil it down to simple elements. This is not going to happen. But we do need to know what the elements are. But then there's still a whole mountain of work to be done, and a difficult task of putting it all back together. And I think that's what we're facing. Same as in genomics, all the A, T, C's, and G's, 3.5 billion of them, do not tell us by themselves a whole lot about what makes us human. And I think in the end, similar gaps will emerge in connectomics, but we don't want to go back to doing science without it. I think this is just one of those things that is beneficial and creates progress, but it is not the answer to everything.
So we're all jointly hoping for something nonlinear in the next
For sure.
I can't resist closing about something without something that has nothing whatsoever to do with this, but has to do with Indiana. I fondly hope that there are no nonlinear surprises in the next election from Indiana.
Please don't mention it. I am very, very apprehensive.
Yeah.
Imagine.
Welcome to that sentiment. Okay, thank you very much, everybody. And next week we will have La Palme. We'll be speaking in French, and it'll be about language.
Okay, thank you. Bye-bye.